important requirement, a biological tool for this technology is the vehicle DNA. Vehicle DNA, also called vector DNA. Vector DNA, also called cloning vector. Cloning vector. A vector DNA is a DNA molecule. It's a DNA molecule that can carry the desired gene into a host cell. And this DNA is capable of undergoing replication in the it should be capable of undergoing replication in the host cell. Host cell most often will be the bacterial cell, or sometimes maybe yeast cell. The vector DNA must also be capable of producing multiple copies of itself, as well as the desired gene. So it should undergo replication, and it should also allow the replication of the desired gene. The vector DNA should have the following characteristics. Number one, it should possess a specific sequence that functions as the origin of replication. Remember, we have described this region in the replicating DNA as ORI. So it should have a specific ORI from where the replication process begins. Number two, it should have a set of antibiotic resistant genes it would have a set of antibiotic resistant genes that are resistant to antibiotics like ampicillin, caramycin and so on. So for resisting antibiotics, like ampicillin or tetracycline or caramycin, because these genes are important to allow the growth of the transferments, that is the cells that have incorporated the foreign DNA, allow the growth of transferments. So this region, these regions are called selectable markers. Selectable markers. And another important characteristic of the cloning vector is that it should have multiple cloning sites. These sites can allow action of several restriction endonucleases. So these are regions that contains various palindrome sequences that can be recognized by specific restriction endonucleases. So the most suitable vector therefore is one which has ORI, which has selectable markers and which has multiple cloning sites.